Welcome to this deep dive into the world of genetics and biochemistry. Today, we are exploring a gene that serves as a vital bridge between two of the most important metabolic pathways in the human body. We're talking about the MTR gene. You might know it by its full name, 5-methyl tetrahydrofolate homocysteine methyltransferase. In the world of biology, we often just call it methionine synthase. While it might sound like a mouthful, this gene and the enzyme it produces are responsible for a chemical handoff that keeps your cells healthy, your DNA protected, and your heart functioning properly. By the end of this video, you'll understand how the MTR gene works, why it requires vitamin B12 to function, and what happens when genetic variations change the way it behaves. To understand MTR, we first have to look at the big picture of cellular metabolism. Our bodies rely on two interlocking gears known as the folate cycle and the methionine cycle. The MTR enzyme sits exactly where these two gears meet. Its job is to take a methyl group, which is just one carbon atom and 3 hydrogen atoms, from the folate cycle and move it over to the methionine cycle. Specifically, the MTR enzyme catalyzes this reaction. On one side, it takes 5-methyl tetrahydrofolate, the active form of folate, and turns it back into tetrahydrofolate, which the body can reuse. On the other side, it takes an amino acid called homocysteine and converts it into methionine. This might seem like a simple trade, but it is one of the most consequential reactions in human biology. If MTR stops working, the entire system grinds to a halt. Why is making methionine so important? Methionine is an essential amino acid, but its most critical role is serving as a precursor to a molecule called SAM, or S-adenosylmethionine. SAM is known as the body's universal methyl donor. Think of SAM as a delivery truck carrying methyl groups. It travels around the cell and donates these methyl groups to various targets, including your DNA, proteins, and neurotransmitters. This process is called methylation. Methylation acts like a series of on-off switches for your genes. It allows your body to silence certain genes and activate others. Without the MTR enzyme creating a steady supply of methionine, your ability to regulate your DNA through methylation would be severely compromised. The MTR enzyme doesn't work alone. It has a cofactor, or a partner, that is absolutely required for it to function, vitamin B12, specifically in the form of methylcobalamin. Inside the MTR enzyme, there is a cobalt atom. During the chemical reaction, the methyl group from folate is first transferred to the B12 molecule, creating methylcobalamin. Then, the B12 passes that methyl group onto homocysteine to create methionine. This is why a B12 deficiency is so dangerous. Without enough B12, the MTR enzyme becomes inactive. When this happens, a phenomenon occurs called the methyl trap. Folate gets trapped in its 5-methyl form because there's no MTR enzyme available to take the methyl group away. Even if you have plenty of folate in your system, your cells can't use it for other things, like making DNA. This is a primary reason why B12 deficiency leads to megaloblastic anemia. Your cells simply can't divide properly. Now let's talk about the gene itself. The MTR gene provides the instructions for building this enzyme. It is located on the long arm of chromosome 1. We all have the MTR gene, but we don't all have the exact same version of it. Small changes in the DNA sequence, known as single nucleotide polymorphisms, or SNPs, can change how efficiently the enzyme works. The most well-studied variant in the MTR gene is known as A2756G. In this variation, the DNA base A, adenine, is replaced by G, guanine, at position 2756. This results in a change in the enzyme structure. The amino acid asparic acid is replaced by glycine. Research into the A2756G variant is ongoing. Some studies suggest that the G version of the gene may actually increase the activity of the enzyme, meaning it processes homocysteine into methionine faster. While faster sounds good, biology is all about balance. If the enzyme is too active or not active enough, it can shift the delicate balance of the folate and methionine cycles. Why should we care about how well the MTR enzyme is working? The biggest reason is homocysteine. Homocysteine is a natural byproduct of metabolism, but it needs to be kept at low levels. If the MTR enzyme is sluggish, either due to a genetic variant, a lack of B12, or a lack of folate, homocysteine begins to build up in the blood. This condition is called hyperhomocysteinemia. High levels of homocysteine are linked to several health concerns. Cardiovascular health. Elevated homocysteine can damage the lining of the blood vessels and promote blood clots, increasing the risk of heart disease and stroke. Neurological function. Because MTR helps produce neurotransmitters through methylation, its dysfunction is being studied in relation to cognitive decline and mood disorders. Prenatal development. Like the more famous MTHF gene, MTR plays a role in neural tube development. Proper methylation is essential for a growing embryo to form its spinal cord and brain correctly. You may have heard of the MTHF gene, which has gained a lot of attention in recent years. It's important not to confuse the two. While they both work in the same system, they have different jobs. MTHF is responsible for creating the 5-methylfolate. MTR is the enzyme that actually uses that 5-methylfolate to convert homocysteine to methionine. They are like two workers on an assembly line. MTHF prepares the part, and MTR installs it. If either one of them is struggling, the whole assembly line slows down. This is why many people who look at their genetic methylation profile look at both genes together. To wrap things up, the MTR gene is a fundamental piece of our metabolic machinery. By producing the Methionine Synthase enzyme, it ensures that homocysteine levels stay in a healthy range, the body has enough methionine and SAM for DNA methylation, and folate is recycled so it can be used for DNA synthesis. Its absolute dependence on vitamin B12 makes it a perfect example of how our genetics and our nutrition work hand in hand. Whether you have a common variant like A2756G or the wild-type version of the gene, the best way to support your MTR enzyme is by ensuring adequate intake of B12, folate, and B6. Understanding your MTR gene isn't just about knowing your DNA, it's about understanding the complex, beautiful chemistry that keeps you alive every single day.